Welcome, everyone. I'm excited to introduce a very interesting speaker today. So Dr. Ted Satterthwaite is an associate professor in the Department of Psychiatry at the University of Pennsylvania School of Medicine. So he completed his medical and graduate training at WashU in St. Louis, working with Randy Buckner, and then joined the faculty in the Department of Psychiatry at the U of Pennsylvania in 2014. So since 2019, he's directed the Penn Lifespan Informatics and Neuroimaging Center. And so basically, Ted uses multimodal imaging approach to understand both normal and abnormal patterns of brain development in the context of neuropsychiatric illness. His work has been very well funded by the NIMH and recognized by several research and teaching awards. And today, Ted is going to share some exciting work with us on the development of the association cortex in youth. And Ted, we'll turn it over to you now. So you can go to your typing side.
Great. And thanks for that really kind introduction. I'm sorry I can't be there with you guys in person, but I really appreciate the invitation, and I'm excited to tell you about some of the work we've been doing recently. I think it aligns with a lot of the awesome work going on in your center, which even though I study kind of the earlier part of the lifespan, I think there's a lot of similarities, which we'll get to. So I'm a psychiatrist, and the clinical context of this is what got me started on this line of research. First is that psychiatric syndromes are incredibly common. Around 1 out of every 2 people will experience impairing symptoms at some point in their life. And unlike other major mental health problems like cardiovascular disease or neurodegenerative conditions that begin late in life, major mental illness starts really young, usually in childhood, adolescence, or young adulthood. And based on this early age of onset, as well as evidence for both basic research and translational models, psychiatric conditions are increasingly understood as neurodevelopmental disorders. Accordingly, the overall goal of my research is to understand how major mental illness is encoded in terms of abnormal patterns of brain development. However, to understand abnormalities associated with mental illness, we first need to understand how the brain develops normally, because understanding the timing of how specific brain systems develop may help us understand the emergence of symptoms. And the main overall theme that I can try to convey here is that to understand brain development, one also needs to understand fundamental properties of brain organization. And one incredibly useful frame of reference goes back to now classic work by Marcel Myelin, who described cortical organization according to its local anatomy and circuit properties. So Myelin showed that the brain is organized into large-scale neurocognitive networks that support hierarchical information processing. This goes from primary cortex here in blue, that it has a highly defined layer structure and canonical feed-forward circuits, all the way up to transmodal association cortex here in pink, with less defined layers and non-canonical circuits. Since Myelin's work, imaging technologies have advanced dramatically and allowed us to understand the anatomical hierarchy in unprecedented detail with things like cortical myelination and other measures. Notably, this anatomical hierarchy aligns really closely with an evolutionary hierarchy that indexes the degree of cortical expansion from primates to humans. And what's remarkable is that these two hierarchies really show incredibly tight convergence, with primary somatosensory cortex being highly myelinated and showing limited expansion compared to primates, whereas association cortex is highly expanded evolution and lightly myelinated. So a recent project led by a truly phenomenal graduate student in the lab, Valerie Sidner, went to look at many more feature types beyond anatomy and evolution. And what Val found was that variation in diverse macrostructural, microstructural, functional, metabolic, and transcriptomic features also recapitulate the cortical hierarchy in ways that we see in anatomy and evolution. Looking across these feature types, what one sees is there's a very clear axis of brain organization that spans from sensorimotor cortex to association cortex. And I refer to this as the sensorimotor-to-association axis, or the SA axis for short. And for the rest of the talk, all the data I'm going to show generally supports a single take-home point: that brain development both aligns with and shapes this axis of brain organization. To put it another way, there's a spatio-temporal hierarchy that in part governs development and leads it to march up this axis in a systematic way, with the association cortex undergoing the most delayed and protracted development. And this pattern of development ultimately gives rise to the cortical hierarchy we see in adults and may have implications for how the brain ages normally and abnormally in the context of neurodegeneration. So nearly all the data I'm going to show today is from functional connectivity MRI, which, as many of you are familiar with, is simply a time series correlation between the fMRI signal at different parts of the brain. Now, functional connectivity is a powerful tool to study the development of brain organization because, as shown by incredibly impactful work by Daniel Margulies, Johnny Smallwood, and others, functional connectivity encodes a clear principle and gradient that aligns quite closely with the sensorimotor-to-association axis and other brain features. This principal gradient of functional connectivity captures variance in network architecture, the spatial ordering of networks in the cortex, and importantly, diversity in cognition and behavior. Furthermore, functional connectivity allows us to define brain networks. And if you look at canonical brain networks that are now like 10 years old, like this one from Jonathan Power, and this one from Thomas Yeo and Randy Buckner, there's really substantial convergence across efforts from different groups. And one important facet is that while these sensory motor networks, like this one in blue, tend to be very spatially compact, functional networks in association cortex, such as the frontal parietal system here in yellow, or the ventral attention system here in this bright purple color, they're really spatially distributed. And today, most of the work I'm going to present focuses on the development of these large-scale, spatially distributed association networks. The work on the development of functional networks builds an incredibly rich literature that establishes that association cortex undergoes dramatic and protracted development in youth. For example, this canonical study from David Van Essen's group demonstrated the expansion of the cortical surface area between infancy and adulthood parallels to a remarkable extent that expansion of the cortical surface between humans and macaques in evolution. Notably, both evolutionary expansion and this sort of structural brain development aligns quite closely with the sensorimotor-to-association axis. So at this point, it's natural to ask, what's the function of association cortex that has expanded so much in evolution and is refined in development? And convergent evidence from many different groups suggests that association cortex is largely responsible for the vast array of behaviors and cognitive processes that really make us human. And today I'm going to be focused on one broad domain of cognition that's particularly important in adolescent development and important for psychiatry, executive functioning. Now, executive function is a broad domain of cognition that encompasses many different cognitive processes, including working memory, set shifting, response inhibition, and other things. And here you can see that executive tasks like working memory strongly recruit association cortex and directly line up with the sensorimotor-to-association axis. Furthermore, unlike other cognitive domains such as episodic memory or motor function, executive function undergoes a really protracted period of development throughout childhood and adolescence and into young adulthood. And this gets us to another reason we should care about executive function, which is that all the behaviors that get adolescents into trouble, think about things like drug use, violence, risk-taking behaviors, teenage pregnancy, yada, 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 they're associated with deficits of executive function and failures in them. And also as a clinician, I know that nearly every major neuropsychiatric condition is associated with deficits of executive function. For example, a fantastic research track psychiatrist in the lab, Sheila Shanmugam, found that regardless of what a patient's underlying diagnosis was, the overall level of transdiagnostic psychopathology was associated with a reduced ability to recruit association cortex during working memory task. So just to recap here, convergent features for many data types show that the brain is organized into a sensory-to-motor-to-association axis. We can study this using fMRI, as the principal gradient of functional connectivity encodes this axis. In particular, fMRI allows us to look at distributed association networks. And so we're going to build on earlier work that looked at structural development that showed that the development of brain structure lines up with the sensory-motor-association axis. And in particular, we're going to focus on the maturation of executive function and examine how this may support the development of executive function and dysfunction in the context of psychopathology. And so the data I'm going to show you really ties to two related questions. How do brain develops normally? How do brain networks develop normally in youth? And how do developing association networks support executive function? To address these questions, I'm really going to present two lines of research, two recent studies, and then end with a bit on some of the tools we use for this sort of research. And I want to say at the beginning that all the work I'm going to show you relies on just incredible trainees in the lab, but also amazing collaborators here at Penn. And without these collaborators, really none of the work would be possible. I especially want to give a shout out to Danny Bassett, network scientist, Young Fan in machine learning, Taki Shinohara for imaging statistics. Also, all the data I'm going to show you comes from one data set that was collected at Penn, the Philadelphia Neurodevelopmental Cohort. It's a large-scale imaging study, including 1,600 kids who were scanned on a single site within 2 years. It was led by Raquel Gurr and Hakan Hakanarsen, along with Ruben Gurr and Monica Calkins. So the first study I'm going to show you focuses on functional topography of association cortex in youth. And this is awesome work done by former postdoc Zai-Shu Cui, who's now a PI at the Chinese Institute for Brain Research in Beijing. And it was really motivated by another critical feature of the Sensorimotor-Association axis, that cross-person variability tends to be higher in association cortex and lower in sensorimotor cortex. This has been documented in many features, such as functional connectivity, sulcal depth, and proportional surface area. Now, this cross-person variability turns out to be particularly important for studies of association networks. And typically, when we use canonical network templates, we assume that by normalizing the structural image to an anatomic template and applying it to a co-registered functional image, we've also successfully lined up functional brain networks. However, there's a lot of evidence now that suggests that this is demonstrably false, and this kind of one-size-fits-all approach for functional brain networks is missing a lot. And just to explain that a little further, a first study to show this came from Tim Laumann, Russ Poldrack, and Steve Peterson. He used Russ's amazing MyConnectome data set to show that a highly sampled individual, Russ Poldrack himself, who was scanned for many, many hours, has many highly reproducible details in his brain networks that differ from a standard group atlas. And moving forward, I could call this distribution of functional networks on the anatomic cortex functional topography. And this study showed that individual specific functional topography really is a thing. People's functional neuroanatomy is different. It can't be fully explained by their anatomy. Now, Evan Gordon, Nico Dosenbach, and others built on that N of 1 study using 10 highly sampled participants from the Midnight Scan Club and showed that basically every single individual they looked at had their own functional topography. They had their own layout of functional networks on their anatomic cortex. Now, over time, it's become really clear that variability in functional topography is itself variable across the cortex. In fact, just like we've seen with other data types, variation in functional topography is maximal in association cortex. And this is exactly shown by these two independent studies, one from Hesheng Liu's group and the other from Thomas Yo's group. And what it means is the physical layout of functional networks on the anatomic cortex varies between people. And this variation is highest in association cortex. However, it's important to note that in all these studies, they were conducted in adults. It really remained unknown how functional topography develops in youth and how it might support executive function in kids. So that's exactly what ZyShoot looked at. And to do this, we used data from the Philadelphia Neurodevelopmental Cohort and created personalized functional networks using a recently developed machine learning technique that uses a method called spatially regularized non-negative matrix factorization, or NMF. This technique was developed by our collaborators, Hongming Lee and Yong Fan. This previously was validated using data from the Human Connectome project, as you can see here. So here, what Hongming showed is that networks derived from resting state fMRI aligned really, really tightly with task activation patterns from the same participant. So we use this technique to build individual specific networks in 693 participants in the PNC who had 27 minutes of high quality fMRI data. Now, one challenge in studying individual specific networks is that if you give a machine learning algorithm too much freedom, you may lose the ability to find correspondence across people, which you need for data analysis. So to ensure there's correspondence that everyone had the same networks generally, this method first requires that we build a group atlas that then gets subsequently tailored to each individual's brain. So here we specified 17 networks to allow comparison with other methods. And they mainly pull out things that we recognize, like the default mode networks, frontoparietal networks, somatomotor networks, etc. And so if you look at this in an individual participant, what NMF does is it yields a soft probabilistic parcellation for each network, which we can discretize for display. And if you examine results across individuals, you can see immediately there's a tremendous amount of variation in the functional topography of association networks between individuals. These networks are in different parts of the anatomic cortex depending on who you look at. Now we can look across the whole sample and we see, just like in studies in adults, the variability in the layout of these networks and their functional topography is highest in association cortex. So just like in adults, association networks in kids have the highest variability in where they are in their functional topography. And this variability map reminded us immediately of other features of the sensorimotor-association axis. So we examined directly whether variability in the layout of these functional networks was correlated with other major features of the sensorimotor-association axis. And to do this, we use really conservative spin-based spatial permutation tests. What we found was that the variability in functional topography was positively correlated with evolutionary expansion. So networks that expand more in evolution are more variable across people. It's negatively related to cortical myelin content. So the more heavily myelinated a network is, the less variable it is. And then it's also related to metabolic costs. So more variable networks also get more cerebral blood flow. So as a next step, in order to examine how functional topography was related to things we care about, things like age in kids and executive functioning, we first derived a super simple summary measure to measure this high dimensional data. What we came up with was a measure called the total network representation. And the total network representation simply quantifies the amount of cortical real estate given to each network. So for example, you can see here a participant with low total network representation of the frontoparietal network here. And in contrast, you can see a kid with a bigger frontoparietal network. And notably, since all these measures are calculated in normalized template space, we're not aliasing in things like different size brains or different surface area here. It's all just the functional networks. So as a next step, we looked at how this measure of total network representation, the amount of cortical real estate given to each network, is associated with age and executive function. And somewhat to our surprise, we found that the total representation of every network did not change with age. However, we found that the total network representation of a couple networks was associated with executive functioning. And specifically, we found that kids with better executive function had a greater representation of two frontoparietal networks. And this makes sense because we know that those frontoparietal networks are the ones that subserve executive function. And so what we see here is that kids who have more cortical real estate given over to the networks that are responsible for executive function do better at executive tasks behaviorally. So while these are really cool results, we wanted to move beyond that very simple summary measure and capitalize on the full richness of the high dimensional data. And so NextISU used a ridge regression model to predict executive function using split half cross validation and nested parameter tuning. So training the model on half the data and the testing on a totally unseen half. And what we found was that we could predict executive function in unseen data with a pretty high degree of accuracy, correlations above 0.4 in both halves of data, and with significance well beyond the precision of permutation tests. And while this was cool, and we knew that like the overall real estate of networks wasn't changing with age, we also tried this with age just to see whether it would work. And we didn't expect it to work given the previous results. And so to our surprise, we found that we could actually predict age even better than executive function, correlations above 0.6 in both halves of the data. And this was initially confusing because we knew that these networks weren't expanding or contracting as kids grew up. And so after a lot of more data analysis, basically what we found was that even though the overall size of the networks wasn't expanding or contracting, what's occurring is that the network borders are being refined and sharpened. And that this sharpening and refinement of network borders is what allows you to see how old a kid is just by their functional network topography. Furthermore, in a recent follow-up study, it suggests that this is not just true for age, it's true for sex as well. In awesome work that's now under revision at PNAS, Sheila Shanmugam demonstrated that functional network topography could allow a machine learning model to accurately identify the sex of an unseen participant. So like age, this was driven by features in an association cortex, the ventral attention network, the frontoparietal network, and the default mode networks. Furthermore, Sheila went a step further in this study and used data from the Allen Brain Atlas to show that locations that allowed us to identify participant sex was enriched for expression of genes on the X chromosome. So here, just to recap, what we found was that functional networks and their topography, where they're laid out on the anatomic cortex, they're more variable in association networks. This aligns with other primary features of the Sensorimotor-Association axis. Furthermore, using a very simple summary measure, we found that kids who have more real estate devoted to the frontoparietal networks that are responsible for executive function are better at executive tasks. This allowed machine learning algorithms to predict executive function with a reasonable degree of accuracy in unseen participants. And to our surprise, we found that we could predict age even better. And this was driven by refinement of the borders of association cortex. So that study was really exciting, but it was really just the first step, and it has motivated two ongoing studies. One is to try to use these individual specific functional networks for targeting TMS. This is a study with Desmond Oates, Stanley Bassett, and Young Fon. And now we're applying these methods to longitudinal data from the ABCD project together with Damien Fair at the University of Minnesota to see kind of a better view of brain development, which is to follow kids over time. All right, so I'll move on to the next section, which features an unpublished project by a fantastic graduate student, Adam Pines, focused on patterns of network development. And I'm incredibly excited about this work, which is currently under review, but also available as a preprint. So in the prior section, I talked about how functional topography of individual specific networks evolves with age. However, one thing that may have occurred to you that we didn't evaluate is how these networks are connected to each other. What is the connectivity between networks, not just the layout of the networks? So here we evaluate that question, and in order to ensure that any results weren't driven by a specific number of networks, like we looked at 17 networks in the last study, instead of just building a single atlas, we used the same machine learning tool to define personalized networks at 29 different resolutions, ranging from 2 networks all the way up to 30 networks. And then we looked at the coupling between these networks. That was our main measure of interest. Simply how connected a given network was to all other networks. And this measure provides a very, very simple summary of network segregation versus integration. Integration, more connectivity with other networks; segregation, less connectivity with other networks. And we wanted to know how this network coupling evolved with age and was related to executive function. And that's basically what we have plotted here. Here, each network at all these different resolutions is a separate line. And on the y-axis, we have between network coupling. On the x-axis, we have age between 8 and 22, or 8 and 23. Furthermore, we've colored each network by its position on the Sensorimotor-Association axis, with transmodal networks being in yellow and unimodal networks being in purple. And if you look at this plot, 2 things are immediately apparent. First is that even at age 8, there's some substantial differences, and unimodal networks here in purple have greater between network coupling than association networks in yellow. Association networks actually tend to be more segregated, while sensorimotor networks tend to be more integrated. Furthermore, as kids grow up, this pattern diverges even more. There's more between network coupling and unimodal networks and more segregation in association networks. But when you put this together, what you can see is that the Sensorimotor-Association axis becomes more distinctly defined with development. You can see you go from here up to here. And if you just replot the same data, another way to see how development coheres with the Sensorimotor-Association axis is simply to plot assigned age effects versus transmodality directly. So here on the y-axis, we have the age effect. So positive values mean you're getting more integration, more between network coupling. Negative values, more segregation, less between network coupling. And on the x-axis, we have each network's position on the Sensorimotor-Association axis, which we're calling transmodality. Also, we've colored each network according to their maximum overlap with Thomas Yeo and Randy Buckner's canonical 7-network solution. So, for example, here in blue, we have somatomotor networks, whereas in red, we have default mode networks. And then here in gray, we have networks that didn't survive, that didn't have a significant age effect after rigorously correcting with multiple comparisons. And what you can see, though, if you just follow the fitted line, is that there's an incredibly clear alignment between how networks develop with age and their position on the sensorimotor-to-association axis. Specifically, lower order networks on the sensorimotor end of the assay axis increase their between network coupling with age and become more integrated. In contrast, the most transmodal networks, such as the default mode and front parietal network, tend to become more integrated as kids grow up. This, it's not an exaggeration to say that this relationship is for sure one of the strongest effects I've ever seen in developmental neuroscience. So, the next obvious question is, well, what are the implications of this striking pattern development for things we care about, like executive functioning? And so as the next step, we looked at the same measure between network coupling and examined how it's associated with executive function while controlling for age. And somewhat to our surprise, we saw pretty different pattern results. Here again, on the x-axis, we have each network's position on the sensorimotor-association axis. So these are more unimodal networks, these are more transmodal networks. And on the y-axis, instead of the age effect, we have the effect of executive function. So positive values means that with more between network coupling, kids have better executive function. So more integration, better executive function. whereas negative values mean the opposite. Better executive function with more segregation. And we found that instead of the linear effect we saw with age, that there's this quadratic effect with more segregation. The extremes of the Sensorimotor-Association axis were associated with better executive functioning. This is really driven by the default mode on one end and somatic motor networks at the other. And if you look at these two plots side by side, you can see that they line up in some ways but are different in others. First, in association cortex, the wind's blown in the same direction. Transmodal networks like the default mode network here in red, they tend to become more segregated with age, and more segregation while controlling for age is associated with better executive functioning. However, if we look at the other end of the sensorimotor networks, we see the opposite. The normative pattern, in fact, is to become more integrated with age, where, however, better executive functioning is associated with the opposite of the phenomenon, more segregation. So because we know that executive function dramatically improves with age, this probably doesn't make a ton of sense to simply control for age like we did in the last analysis, and instead to do a statistical mediation analysis, which examines how much the refinement of between-network connectivity might mediate the known improvement of executive function with age. What we found is that the degree to which between-network connectivity mediates the development of executive function is strongly driven by that network's position on the Sensorimotor-Association axis. Specifically, just like we'd expect from the prior two plots, the normative segregation of transmodal networks, like the default mode, they tend to become more segregated as kids grow up, and that segregation is associated with better executive functioning. In contrast, the normative integration of networks at the sensorimotor end of the axis tends to suppress the development of executive functioning. So if you pull all this together, between network coupling and individualized multi-scale networks, between network coupling and individualized multi-scale networks is higher in sensory motor networks than in association networks, and this divergence increases with age. Specifically, age effects are strongly associated with the network's position on the Sensorimotor-Association axis, with sensorimotor networks integrating, as you can see here, whereas association networks tend to segregate. In contrast, executive function had a distinct pattern, leading to dissociable mediation effects, with normative segregation of association networks being associated with development of executive function, whereas the integration of sensorimotor networks tends to suppress the development of executive functioning. And what is striking is that these results in development parallel to great degree results have been shown in the aging literature. In particular, your guys' very own Gog and Wig showed that sensorimotor integration that we see starting early continues across the whole lifespan. So this is sensorimotor system segregation declining into the 80s here. Furthermore, Goggin showed that network segregation that we see in association networks in development is flat through midlife here, but then also tends to decline in aging. Subsequently, his findings have been replicated in other samples and extended to neurodegenerative diseases, including Alzheimer's disease. These are from two papers in the past 2 years, Chen and Cassidy et al. And so what this suggests is that this normative process of how brain organization becomes instantiated in brain development may unwind in aging and be associated with cognitive decline. So I'll close here to make sure we leave plenty of time for questions and talk about several tools for reproducible developmental neuroscience we use in the lab to try to make sure our work can be easily scalable and highly reproducible. So the first comment is that charting normal brain development takes large samples. If you want to look at abnormal brain development, you're going to need huge samples. However, as you get to very large samples, you have a lot of statistical power to find effects, but that also creates a ton of sensitivity to artifacts. So probably the most notorious artifact in this kind of domain of the literature is motion artifact. And motion systematically biases estimates of functional connectivity. This goes back to 3 studies that were published within 3 months in Neuroimage in 2012, Jonathan Power and Steve Peterson, Kuna Van Dyke and Randy Buckner, and our study here at Penn, all showed the same thing using 3 independent data sets and independent methods. And basically, what everyone showed is the motion artifact is really bad for functional connectivity. Furthermore, it has a lot of structure. So this is a kind of a complicated plot, but I'll walk you through it. Is that if you look at on the y-axis, the correlation between motion and functional connectivity, the more that basically says, if you move in the scanner, what does it do to your measurement of functional connectivity? And you plot it against how far nodes are apart in the brain, so how far what you're trying to measure is. You'll see that brain regions that are close together, as people move more, they tend to get measured as higher connectivity, whereas as people move more, if you're trying to measure distant brain regions, connectivity will be suppressed. What's important about this is that before 2012, this was like kind of the canonical finding in brain development, that nodes that brain regions that are close together tend to get more connected, and things that are farther apart tend to get less connected. But in fact, subsequently when reevaluated, this was probably almost completely driven by motion artifact, which was sad. We've gone on to show that this isn't just about functional connectivity, but you actually see incredibly similar effects in diffusion tractography that measure structural connectivity. They're very similarly impacted by motion. And this matters because if you don't account for this sort of artifact as you study brain development, you're not going to be studying brain development. You'll just be studying motion artifact. So this is a very simple plot that shows 4 different ways of handling motion artifact in your data. And if you like here, this is kind of standard pre-2012 processing, where you do like kind of not substandard denoising and you don't handle things in your group level analyses. And this is kind of what most people do now and what we recommend. And what you see is if you don't carefully account for motion artifact, you'll find 5 times more quote unquote developmental effects. But that 80% of those results will probably be almost completely driven by motion. So subsequently, because of the magnitude of this problem, we've put a ton of effort to try to figure out how best to denoise data. I think probably the most important thing is how to better to acquire data, but having acquired a lot of data as is, we subsequently put a lot of effort into how to best to control motion artifact and denoise it. And work by an awesome data analyst, Roscoe Chidich, who was a data analyst in the lab, is now Russ Poldrack's bioengineering graduate student at Stanford. He looked at 14 different pre-processing pipelines just to see how they did. And what he found was there's incredible heterogeneity in the associations between functional connectivity and motion in these different pipelines. So what you can see is that commonly used pipelines leave up to 80% of your edges in your network that are to be significantly associated with motion, whereas kind of some of the better pipelines, including ones we use at Penn, people at WashU and other people, they will leave less than 2% of your edges significantly associated with motion, even in very large samples. So coming out of this work, Roscoe developed a code base and a software package called the eXtensible connectivity pipelines to allow us to do really top-notch motion control and performance assessment. And Roscoe initially conceived of this as a soup-to-nuts pre-processing pipeline that went from like right at the beginning all the way to the end. However, when we're building this, at the same point, Oscar Esteban, Russ Poldrack, and Chris Gorgolowski at Stanford created fMRI Prep, which does around the first third of what XCP does, and it actually does it just way better. And in the end, I want to use software that works. I don't really care whether we built it or not. And so Aziz and Mbimpe, a senior data engineer in the lab, reconfigured our pipeline to work directly with fMRI Prep and do post-processing of data that has gone through fMRI Prep. This has been a really successful strategy. The software works really well together, and as a result, it's been widely used with over 6,000 unique downloads. Subsequently, we wanted to have this sort of functionality not just for functional imaging, but for all the other multimodal imaging we do in the lab, including diffusion imaging and arterial spin-labeled MRI to look at brain perfusion. So Matt Cieslak, a senior scientist in the lab, created QSI Prep, which is a set of pipelines that auto-configure for diffusion data. And what it allows you to do is handle nearly any diffusion acquisition. And because it's what's called a BIDS app, it reads the imaging parameters from the metadata encoded in your imaging data to build appropriate and fully reproducible pipelines for the data. Furthermore, it includes modular reconstruction workflows that are interoperable, allowing us to leverage best features of individual software packages that often are incompatible unless you use them together in this sort of framework. This was published in Nature Methods earlier this year and has been downloaded over 7,000 times just in the past year. And as part of the evaluation and testing of QSI prep, Matt looked at many different acquisitions, and he compared QSI prep to custom pipelines that were used to process the data for those studies. And what Matt found was that automatically configured pipelines by QSI prep performed as well or better than custom pipelines for each study. Specifically, he found that QSI prep introduced less blurring into the data and it preserved more anatomical detail. Also, while doing this, we had higher data quality than pipelines tailored for each acquisition. Most recently, Aziz Nabimpe, senior data engineer, built ASL prep for processing arterial spin label MRI and allowing us to quantify cerebral blood flow. The paper describing ASL prep is out on bioRxiv and is under revision in Nature Methods. And like fMRI prep and QSI prep, it builds automatic workflows for processing your data using top methods. As part of the evaluation of ASL prep, Aziz processed over 3,000 data sets from 5 studies across the lifespan. And what he found was a striking pattern of evolution of cerebral blood flow, with cerebral blood flow declining in youth and into early adulthood, flattening out in middle age, and then showing a decline again in aging. So these tools have been particularly useful for the work that I'm perhaps most excited about right now, something called the Reproducible Brain Charge Project, or RBC. This is a study I lead together with Mike Myelin at the Child Mind Institute. And RBC is an effort to aggregate, process, harmonize, and share several of the largest studies of brain development, including the PNC, the data set I was talking about primarily today, but also the Healthy Brain Network, which is even bigger, run by Mike in New York, and Kai Rockland sample, his previous study, the Brazilian high-risk cohort, which is an amazing 3-wave longitudinal study of kids in Brazil, and the Human Connectome Development Project. So, and there's also several others. So the total number of samples is over 10,000 samples. And for all these studies, we're carefully curating, QAing, processing all of the functional imaging data and structural imaging data using fully reproducible pipelines. Furthermore, in work led by Giovanni Saloom, the PI of the Brazilian high-risk cohort, we're harmonizing diverse clinical measures from these studies. So hopefully we'll be able to look at not just brain development, but also abnormalities associated with psychiatry, with psychopathology. And importantly, so no one else has to do the really painful work here that we're doing of curating and processing all this data, we're going to share all the raw data, all the process data, all the containers, all the code openly on Indy, which is a freely accessible bucket on AWS. We really hope that it'll allow everyone to use these data sets much more easily and kind of go the route of that kind of has been very profitable in genomics. Discover, replicate across data sets, and really combine data resources in a way that we haven't been able to before. So I'm going to end here and leave time for questions. I want to thank all the people who actually do the work in the lab, in particular Aziz, Matt, Sydney, Zaishu, Adam, Sheila, and Val. Many collaborators in psychiatry, in particular Raquel Gur, who's the PI of the Philadelphia Neurodevelopmental Cohort. Our technical collaborators I mentioned at the beginning, Yong Fan, Danny Bassett, and Taki Shinohara. Damien Farrett, University of Minnesota, Mike Myelin at Child Mind Institute. Grants from NIMH, and in particular, our participants and their families. Thanks, guys.
All right, questions from the room? But speak up.
That's a great talk. Can you hear me?
Yeah, sure can.
So I just want to go back to the one of the data points you had earlier in your talk about the relationship between, as you called it, real estate occupied by network and executive function. So obviously the natural thought there is that the causal relationship is more real estate equals better function. But do you have any reason to think that there's a possibility of either reverse causality or reciprocal causality? That is, it's not just the fact that, you know, more real estate means that you're cleverer, but that being cleverer interacts with your brain to give you more real estate. Do you have any temporal information? Obviously, what I'd like to do is to show that the real estate emerges earlier than the ability, but that's going to be a tricky thing to show, I guess. But do you have any thoughts about that?
No, I think you nailed it. So first of all, if I used any causal language over the past 40 minutes, I apologize, and I would like to strike that from the record.
I'd like to make clear that you did not. I'm not accusing you of that. I'm just saying it's a natural assumption that people might make.
Yes, no, absolutely. Well, I think I should include plenty of caveats, though, because I think one of the frustrations of this sort of research is that we're not performing experiments in the traditional definition. We're not. There's no independent variable that we're manipulating here. I'm not running a clinical trial. I'm not doing neuromodulation. There's not really an intervention. These are simply associations. Yep. And I think they're interesting associations, but the problem is our brains are so well trained for reverse inference that we draw those causal arrows extremely readily. And I think the points you're bringing up are incredibly important, and they're basically impossible to disentangle from this sort of data. I think the important question you asked is, well, if we can't with this sort of data, what sort of data do we need to disentangle that? And I think we. Oh, I'm sorry. Go ahead. Okay. So I think that it's possible we could have some of the data. The first thing is that the fact that we saw that real estate didn't expand or contract much over this developmental period, but rather the borders were refined a little bit, kind of suggests that the boundaries of these networks are laid down very early. And with some collaborators who work even younger in like younger kids, like childhood, they see the same thing. They don't see these these boundaries moving around to a dramatic degree and the real estate shifting around. So one really important question is, well, what lays down these boundaries? What determines where these networks get drawn and how a given cortical column of it will associate more with some sort of functions than with others? And I think there's probably a lot of ways to attack that problem. Imaging genomics is a very exciting one. We have some collaborations that are starting to look at that, and you can imagine that using both common or rare variants, there might be differences in the population that are associated with more real estate being devoted to a network associated with executive function than others. But to really get to the question you're asking is like timing and precedence. We just need to go much earlier. Maybe even to the fetus to see, okay, when are these networks evolving? How do they evolve? And then follow those kids to see how well they do on executive tasks and what happens to them. And that's an insane study that'll take forever. But luckily, that's something that's starting right now with the with the HBCD project, where NIMH and NIDA and others will follow. I think it's something like 10,000 kids from fetus to 10 years old. And so that sort of data, those can take a while to collect and a long time to follow, could have the potential to answer these sort of questions.
Very cool. Thank you.
Sure, go ahead. I don't have a great view of the room, so you guys can just start talking whenever you're ready.
Thank you for a fascinating talk. On that same topic of representation, I'm not very familiar with it. Is it a zero-sum game where having more representation in one network means you'll have less in others? And does that mean that kind of plays into people being better at some abilities versus other abilities?
Yeah, I mean, it's a great question. It's something I really struggle with. As currently formulated, cortical real estate is a zero-sum game, as we've shown here, but I think there's a lot of caveats there. First is that we've calculated this in normalized space, so we're basically taking out any differences in surface area. But we know that surface area is important, and that kids in general with more surface area tend to be better at some things. More real estate in terms of having a bigger pie to divide up is generally a good thing. But yes, in the formulation we've used here, we're kind of fixing the size of the pie, and then everything you carve it up, and it does turn into a zero-sum game. But I think that that's not actually how it works, and that both the divisions in the pie and the size of the pie is changing, and the shape of the pie is irregular, to push an analogy far too far. And so I think that it doesn't exactly answer your question. I think it gets a little bit complicated because we do know in terms of cognitive skills that the G factor, general intelligence, is dominant. and that people who are good at some things actually tend to be better at others. Like that, like the covariance among cognitive domains is much stronger than functional specialization. This is true in psychiatry also, whereas we see there's been a ton of research, and I didn't talk about it today, this wasn't a very clinically focused talk, but the equivalent of the G factor, the P factor, that's like for general psychopathology, how sick you are in some domains is dominant. So if you have one psychiatric illness, it makes you much more likely to have another. And so comorbidity is the rule, not the exception. And so, you know, this actually kind of drives a lot of our conceptualization, because in general, we think that the same things that cause better general cognitive ability, the reverse of that, it might have vulnerability to psychopathology. So if you have good executive functioning, you tend to be protected against all sorts of different psychopathology. And if you have poor executive functioning, you have vulnerability to all sorts of psychopathology. So kind of the thought that we have right now is, and we have some preliminary data from another study that I didn't talk about today, is that if you have less representation of these higher order association networks that are super important for things like cognitive control and emotion regulation, working memory, et cetera, it's going to lead, you're not going to have bad cognition, you're going to have more vulnerability to all sorts of psychopathology. Does that make sense? I think that was kind of a circuitous, rambling reply there.
Oh, yeah, thank you.
I have a question about something probably really simple that I missed. From those across network scatter plots,
yeah.
the non-significant data points, where are they from spatially?
Let me make sure I can, which, was this the first study or the second one?
The first one, the several plot, you have gray, light gray data points from non-significant.
I'm going to reshare it and we'll go back to the slide you're talking about, so I know, so I'm not just completely not responding to what you're asking. Okay, can you guys see this?
Yeah.
All right, great. So let me see. Was it like?
Go. No, keep going. Like it's in the summary slide. Yeah. Like so yeah, that's an example. Yes.
Okay.
The non-significant.
Yeah, exactly. So those. So first of all, most like one of the great things about studying development is that there's just a ton of signal. Like an 8-year-old is super different than a 22-year-old. It's like so much more satisfying to study in that respect than psychiatry, because the signal in psychopathology is just super weak in comparison. So here you see that most brain networks have significant developmental effects over the age range we study, and only a couple of them don't. This is like a minority of them. It's like I think 20% of them. I don't know actually where they seem to be pretty evenly spread out over the range. Maybe some more in the like kind of more association networks. I don't know exactly what networks those are. I'd have to ask the graduate student who did the actual work. But it's a good question, and I should know the answer. So
they potentially span all the networks.
Yeah, exactly. I mean, it looks like we have both association networks and sensorimotor networks here.
Okay, thanks.
Sure. Did that answer your question?
Yeah, it did. Thank you. Do we have any more questions from the room right now? We'll go to questions online now, if not.
Yeah, I have about 5 questions. The first one is from Goggin, who says, great talk. His question is regarding the first set of observations you presented, demonstrating relationships between the topographic size of networks and executive function in kids. As you know, topographic real estate in the brain is going to be zero-sum. So if participants have larger frontal parietal networks, other networks will have to be smaller. Are there any systematic patterns of that trade-off? And if so, do they have a corresponding behavioral cost? Sorry, that was really long.
No, that's a great question, Goggin. Thank you for first, thanks for the kind remarks. And as I think I've said in person before, a lot of this is in part inspired by your work. So it's nice to see it playing out in development too. You know, I'm not sure. I mean, I think that like most of the trade-offs we see are at the other end of the SA axis, but it's not clear there's a strong behavioral trade-off yet. We need to look into it more. You know, in some of the data we have in, let me see if I actually if I have this in the slide deck. Yeah, I think I do actually. Let me reshare here. All right. So this is from a paper. It's now in bioRxiv by Zai-Shu Cui. It's like kind of a follow-up to that first study on functional topography. And basically, here what Zai-Shu is trying to do is relate dimensions of psychopathology to association cortex topography. And basically what we found is that using the complex pattern of topography, we could predict significantly, but I want to emphasize low effect size, each major dimension of psychopathology. And basically what we see is that more psychopathology is associated with more representation of networks at the lower end of the sensory-motor-association axis and less representation of higher association cortex. I think this is the best example we have yet of kind of the behavioral trade-off between sensory motor and association networks in terms of the real estate.
Okay. The next question from our online viewers says, how does QSI prep compare to DTI prep?
You know, I think that the key thing is that QSI prep is a BIDS app. I'm sure Matt would know more of the details of DTI prep because he's like a diffusion imaging guru, but I have not personally used DTI prep. What's nice about QSI prep is that since it's a BIDS app, BIDS is the brain imaging directory structure. It records the metadata of your images in a JSON, basically a text file sidecar to your images. And what QSI prep does pretty uniquely for these pipelines is it reads that metadata, sees what's in there, and then configures to what's appropriate. And unlike basically any other software that's out there, to my knowledge, can handle any image acquisition. Single shell data like the quick traditional DTI acquisition, multi-shell data like what's connected in HCP and ABCD. It even can handle compressed sensing and non-Cartesian, and Cartesian DSI scans. So it's incredibly flexible, and it allows you to use kind of the best tools in combination in a way that you normally can't. Because what's crazy, I mean, you know, I started out in fMRI, and I have the, and so like every time I learn a new imaging modality, I'm like, the grasses can be greener. It's like, you know, I'm sure they've got it figured out better than fMRI over there. Turns out they haven't. Diffusion imaging is like chaos compared to fMRI, that the different diffusion imaging packages have a really hard time playing together. They use different coordinate systems, different naming conventions. And so using kind of the complementary strengths of each package is really hard. QSI Prep has an interoperable framework that allows you to like kind of take best in class measures across different packages and use them together. So though while we haven't, our benchmarking strategy wasn't for a specific package because there's so many different packages, it was rather for each data set. And we assumed that the dataset collector for these large resource datasets would use and did develop pipelines that were tailored to that acquisition. And what basically we showed is that QSI Prep does better, as good if not better, than these tailored pipelines that are tailored for each acquisition. So what you get is flexibility to handle basically any acquisition, and it performs well. Does that make sense?
Yep.
Okay, last question is a two-part question. First part says, how do you define quantified transmodality?
Gotcha. So so two-part question, two-part answer, all right? So in the first, in Adam's study, the one I showed there in the second part of the talk, we used Johnny Smallwood and Dan Margulies' principal functional gradient from their PNAS study. We just took the map off that they published and said, okay, where is this network on that gradient? Going forward, actually, though, from Val Sidner's neuron review, we use kind of a multi-feature Sensorimotor-Association gradient. All those maps are online, too. She tweeted it the other day. But basically, we, instead of just using that one functional gradient, which is tied to one specific imaging modality, we use kind of the multi-feature one that averages across not just fMRI, but all these other different features, including myelination, evolutionary expansion, transcriptomics, yada, yada, yada. But feel free to email me. I can point you to those maps if you want to use them.
Okay, and to what extent should I assume all prior development studies are invalid because of the movement effects you describe now? Was data you presented today analyzed with that confound movement taken into account?
Yes. Well, I think Any imaging study on individual differences before 2012 using functional connectivity should wonder. There's a great study by Josh Siegel and others at WashU from 2019 that looked in the Human Connectome Project and just say, okay, well, what is associated with motion? Like, and the answer is basically everything you care about. Psychopathology is, age is, BMI is, cognitive scores are. Basically, any study of individual difference, you have to worry is going to be confounded by motion artifact. And the step 0 in that is simply plot your measure versus motion. And if there's no relationship between your measure of individual difference that you care about and motion, then you're okay and you don't have to worry. However, almost always there is. The relationship with age is like 0.3 or something like that. BMI actually is even stronger, 0.4. And in those sort of cases, you really have to worry. And so that's why, to answer your question, yes, that's why we did that really incredibly tedious benchmarking study and looked at 14 different pipelines, is to try to decide, okay, well, which one of these gets rid of the motion of the data while preserving network structure in terms of like being able to see these functional subnetworks. And then, so we use the best denoising, and those results, that was on our data, but those results have been largely replicated by other efforts as well, including a study by Lyndon Parks and Alex Fernito. But then we kind of take the next step, and we also include motion as a covariate in all our group level analyses. So there's really, if anything, people will, on review, will say we're overcorrecting and we're throwing out too much baby with the bathwater. I think given the history and the literature, I'd rather be conservative. And also we have just so much statistical power in these samples that I think we can afford to be conservative because this artifact is really pernicious.
I just weigh in there. There's another issue with motion as well, which is important. I think, which is motion independently predicts cognitive function. So I think Randy Buckner was one of the first people to point this out. In our lab, we consistently find that people who move more in the scanner have lower levels of cognitive performance.
Totally.
Particularly on in-scanner memory tasks, but it extends to neuropsych test scores outside of the scanner as well. So, absolutely. And that in itself is an interesting issue when you start to use motion as a covariate, when you're looking at imaging versus behavior relationships.
Yep.
Because and I haven't really got my head around what that's doing to those relationships, given that motion is an independent predictor of cognition.
Totally.
So it's really, I'm sure you must find that with the kids as well. It's probably the same thing.
Oh yeah, absolutely. You see that very strongly in this data also. And I think there's a couple different approaches to that. I mean, you could, the ideal world, you acquire better data always. Like go back and get rid of that relationship, or get some sort of stratified sample so you're having variation in cognition that's unrelated to motion, or use some sort of or like acquire so much data that you're willing to match and just throw out a third of your sample so there's no relationship. But then once you go that route with matching, then you're like, okay, am I studying abnormal or some weird sample with high people who perform well in cognitive tasks or move abnormally, and these people who are less able at those tasks who are abnormally still, and is it generalizable? Okay, that's a problem. I think that our approach is to acknowledge they're correlated and to go vary for it. And usually with good denoising, it doesn't suck that much variance out of the data. But to the degree it does, I think we just accept that and overcome it with statistical power with large samples. Because I think the alternative of type 2 error in this case scares me a lot less than type 1 error with motion. And we know that, especially historically, there's just been a tremendous amount of type 1 error, false positives that are on development that are really motion artifact. That makes sense?
Yes.
Yeah. Do we have any more online questions, Camilla?
No more questions.
Anything else from the room? Okay. Thank you so much, Ted, for a fantastic talk. Appreciate it.
It's really fun to see you guys. I hope I get to meet you guys in person. Apologies for not being there today, but thank you again for the invitation. It's really kind.